Hi, I'm Sandi Genovese and welcome to the Ellison Education video series. We're looking at a chart that's used to represent meiosis, which is a process by which cells in living organisms reproduce. We're going to show how using the same dye can be used to create a chart for meiosis and for mitosis. So if you look at the table, you can see I've got both of the charts set up here. These were all done with just these two dyes. It's pretty amazing. This is the SureCut, the Ellison SureCut dyes. One dye just has the 4 daughter cells on it, and then the other double cut dye, it shows, if you start here at the top, a cell. This would be like the parent cell, and right here the nucleus for the cell. Here are representations of the chromatin, and then the spindle fibers. Here's chromosomes. Here's the sister chromatids. Then you have the cell elongated and eventually splitting into 2. All of this is amazing because it helps you to create charts and graphs and things that allow every student to be able to create either a chart like this or even flashcards. In order to do it, you need to cut the parts first. So I'm going to take the die, because I'm cutting it in a Big Shot Pro machine, I'm going to turn it so that the blade is up. Now, looking at the chart, you'll see that the colors that I'm going to need is black and white and yellow and blue and red. So I have all of those together and I can cut them all at once. I'm just going to place them over the blade, and then I just need the cutting pad, which I'll put over the top of the whole thing, and then I'll just roll it through the machine, and that's going to cut so many parts and pieces. Look at all the parts and pieces and all the various colors that you have to represent everything you need to create charts. And I'll bring this over here, and you can see there's just all of this. And there's are multiple layers here from this cutting just the one die. Now, if you want to see how easy it is to actually create one of the parts, starting here, the parent cell in meiosis, if I take, and I'm going to represent the nucleus, and notice that I've shown the nuclear membrane along with the little openings that's going to reference that eventually that membrane will disintegrate. This is a very easy thing to do, and then the chromatin are just going to get glued inside. I have them sitting here, so if I just take the chromatin and move them out of the way, and then take the nucleus just with a black marker, all I have to do is seat this so that I'm using it to go along the edge, and I'm going to go a ways and I'll put a little separation there, go another ways, another separation, continue. You can see how easy it's going to be. I'll bring this whole thing over and show how quickly you could put together a parent cell. When you look down at the other phases, every phase is created using all those same things on the die. You've got the spindle fibers, you've got chromosomes, you see the crossing over of the chromosomes, the splitting. Everything is just so easy to do using the dyes. You know, whether you and your students make a chart or you make flashcards, it's going to help them understand the difference between blue jeans with a J and blue eyes that are inherited through their genes, and that's genes with a G.